In this module, we will discuss another important area in the molecular biology, and that is the deletion analysis. What is the deletion analysis? And we'll explain it through the experimentation, that what were the experiments made to establish the deletion analysis. What happened that in 2002, a large consortium of investigators, a large group of scientists, which were led by Ronald Davis, they actually found and reported that the set of yeast mutants they have generated. They generated a set of yeast mutants, and in each of one mutant, one gene had been replaced with an antibiotic resistant gene. So what they did, they took a large number of yeast mutants in which one gene that was replaced with antibiotic resistance gene, and that was flanked by 20-mer sequences, that is the different for each replaced genes. So what happened? That in the molecular barcode, each gene replacement has a molecular barcode. Each gene which was replaced, that has a molecular identification, so it can be uniquely identified. And these investigators, they, as a matter of fact, replaced over 96% of the annotated open reading frame, ORF, in the Saccharomyces cerevisiae. And these investigators, they found that with the barcode, 96% of the annotated open reading frames, ORFs, in Saccharomyces cerevisiae, they were replaced by these. Now, mutants were examined for ability to grow in a mixed culture under different conditions. So these mutants, they grow them in a mixed culture under the different condition of media, temperature, and things like this. And the 6 different conditions which they performed this experiment was high salt concentration, presence of sorbitol, galactose, grown them at pH 8, minimal media, and antifungal agent, that the nystatin was present in some of the media put in that. And these were the 6 different conditions in which the experiment was performed. Now, gene expression, then they were examined under each of these conditions by hybridization of RNA to oligonucleotide microarrays. So the microarray technique was used, and RNA oligonucleotide microarray, they were hybridized under the conditions, and they found that what is the story. To do this genomic functional profile, Davis and colleagues, they grew a mixed culture of all 5,916 mutants under each of the conditions. So remember, such a huge number of mutants they grew, and under these conditions. They collected cells at various times, tested for each barcode by hybridization to an oligonucleotide array. So they tested that each barcode and hybridization to the oligonucleotide array which was put on the slide microscope, and the rate at which the mutant disappears should correlate with the importance of the deleted gene. That is, if the gene is very important, then that mutant should disappear of the deleted gene in dealing with the condition. So in certain conditions, say in PH6, a gene was deleted, was not present, then its growth would be different, and so on, so forth. So they studied the yeast metabolism of galactose. Several genes were found that were known through years of study to be involved in the yeast metabolism of galactose by Saccharomyces cerevisiae. And they found that the 10 new genes that had previously not been implicated in the galactose metabolism, they were also found. So this study resulted in the finding of 10 new genes which were implicated or which were just used or involved in the galactose metabolism were also found in their experiments. Now, the wild-type yeast, they found that there were 11 mutants important in galactose metabolism were tested individually again in the wild-type yeast. They took the wild-type yeast and found that the 11 mutant, they were involved in that metabolism. If you see in this diagram, you will see that there are different mutants and their growth rate is different. For example, with 100% galactose, with very minimum quantity of galactose and all these. And in a very simple way, by demonstrating the growth rate of the wild types of these yeast mutant, they demonstrated that how the growth curves of various mutant that was discovered and profiling to the deficiency in the response to the galactose. It means that they had a deficient galactose gene in the different mutant to the different extents. So that's another important factor and an important evidence that how deletion analysis that can be explained through an experiment, through the simple experiment to determine that where the gene has been situated on the chromosome.